Okay. Just enough time to get a few of these intro slides done and out of the way. Most of this stuff is going to be pretty straightforward. Did you guys do lymphatic in the lab? Sometimes we do, sometimes we don't. Did we do it? Did we skip it? We may have skipped it. There's not a whole lot to do in the lab because we don't have models that show the lymphatic system, and we have a couple of slides. You might have looked at the only things that I asked them to do in the lab. You might have looked at the tonsils, the different tonsils, and the I feel like there's something else. Maybe not. Okay, so what's going on with the bullet points in this slide? Does anybody have questions about the functions of the lymphatic system? This and this are the big ones, okay? Defense and fluid balance. We're going to talk mostly about defense in here. We're going to spend most of our time talking about the immune system. And I will tell you right now, this is my least favorite system. Partially because there's just so much and we can't cover it all in the time that we have. So a lot of what you're going to hear me say is we're going to learn this at this level. If you want to know more, if you're excited by this chapter, good for you. If you're excited by this chapter, Dr. Buck teaches an entire semester of immunology. Covers all of the things immunology related. It's a great class. So you need to know definitions here. You need to know the names of the structures, those sorts of things, just like all of the other systems. All right, so this is still anatomy stuff, right? So you need to understand what lymphatic vessels do, and what is going to cause the contraction of those lymphatic vessels. So not so much what's going to cause the contraction of the vessels, but what the purpose of contraction is, right? So in order to move lymph through the lymph vessels, we have to have contraction of the smooth muscle around the lymphatic vessels, as well as contraction in the skeletal muscle around those lymphatic vessels. And then changes in thoracic pressure, which is basically related to your breathing, and we'll talk about that in more detail when we get to the respiratory system. Make sense? You guys with me? Okay. We are going to talk a lot about B cells and T cells. So I've got my little doohickey here. So we're going to talk a lot about B cells and T cells here in a few minutes. And you need to know those. They're going to be really important in terms of what I'm going to expect you to know about the lymphatic system. White blood cells that come from bone marrow and grow up and they hang out in other places. That's what we're talking about here, okay? There are collagen and reticular cells and other things that act as sort of a trap, right? So when you have bacteria and microorganisms, those sorts of things, they get caught by the tissues in your lymphatic system. And then you have the nodes that are encapsulated tissues. So your lymph nodes, your spleen, and your thymus are all encapsulated, meaning that they have a coating around them. They're enclosed by something. And then the malt lymphoid tissue, which is just beneath the epithelial tissue in your skin, that's not encapsulated. It's just tissue. It's not surrounded by anything, okay? Just need to kind of know what they are, just definitions and stuff. Hopefully you guys took a look at the tonsils in lab. If you didn't, you should. Okay. You should know the palatine tonsils. You should know that the the adenoids, the pharyngeal tonsils, and then you should know that there are lingual tonsils. There are 3 different groups of those. You should know what a lymph node is and what it does. Try that again. That's its job. Okay. There are superficial ones when you go to the doctor and they're feeling around at your throat. That's what they're looking for, superficial lymph nodes, to see if they're swollen in any way. Okay. There are deep ones that are harder to find. Last in the fall, was it in the fall or was it in the summer? Might have been in the summer. I don't know if I told you guys about this. My mother was diagnosed with non-Hodgkin's lymphoma. She's fine now. She did 16 weeks of chemotherapy and PET scan says she's all clear. So all is well. But the reason that she went to the doctor to find out what was going on was because she had a lymph node underneath of her arm that had swollen up to the size of a baseball. And then just before they diagnosed her, she had another one right behind her clavicle that also became physically visible. Okay, those would be superficial or deep. Those would be superficial, right? Superficial, closer to the surface. When they did the PET scan, they found what they called a subcarinal lymph node that also was affected. Now, you haven't learned this yet because we haven't done the respiratory system yet, but when your trachea comes down and it splits to go to your lungs, that split is called the carina. Subcarinal would be below the carina, right? It was also inflamed. It was also affected by the lymphoma, but it wasn't visible because it's a deeper internal lymph node. Does that make sense? Yeah? Okay. There are afferent and efferent lymphatic vessels. This should make you think back to the nervous system. We talked about afferent and efferent with the nervous system. Afferent means something is what? Well, so sensory is particular to the nervous system. But what is afferent? I told you to associate afferent with a particular word. Not away, arriving, right? So things that are coming in through the afferent vessels. And efferent, I told you to associate with exiting, right? So things that are leaving the lymph node are going out through the efferent vessel. You're going to see afferent and efferent again when we get to the urinary system. This is common terminology, and they mean the same thing in every system. Okay. The spleen, located on the left superior side of the abdomen. Okay, on the left, towards the top. It is a very delicate little organ. And if you get damage to your spleen, it is entirely possible that you can bleed to death. The spleen is a very delicate little thing. Its job is to destroy and break down red blood cells. It also will help to detect some foreign substances, and it is a small reservoir for blood. This is why it's so easy to bleed out. Okay. Thymus, little tiny guy. Okay. He's little tiny guy, right? Just at the superior surface of the heart there, in the mediastinum. The first year of life, it starts to get bigger. After that, it stays the same size until you turn 60, and then it starts to shrink. So in the first year, it grows. For most of your life, it stays the same size. Once you turn 60, it starts to shrink. This is the place where your T cells grow up. Okay. Anybody want to make that connection? Why do we call them T cells? Because they come from the thymus, right? Okay. The thymus secretes a hormone called thymosin. Thymosin is important in T cell development. Okay. We're going to talk a lot about T cells. Lots and lots. Okay. Question? They do. T cells mature in the thymus. We'll get there. Hold that thought. Yes. Immune function? Yes. Yes. A splenectomy, removal of the spleen, damage to the spleen can affect your immune system, will affect your immune system. Okay, so we are primarily going to talk about the immune system and immunity for the rest of this lecture, okay? We're not going to spend a lot of time talking about the lymphatics specifically. We're going to talk about the major function of the lymphatic system, which is immunity, okay? So when we are talking about immunity, we're going to use some specific terminology. A pathogen is anything that causes a disease, right? A pathogen can affect any number of organisms, including humans and animals, okay? The immune system, which, and I haven't had this conversation with you guys yet. So if you remember back to A&P 1, way back in the beginning of the semester, the very first lecture, there was a section of your textbook that talked about the 11 systems of the body, right? Immune is not one of them. There is a lymphatic system, and within the lymphatic system, there is a subsystem that is the immune system, right? You're going to see this again when we get to the cardiovascular system, and we talk about the 3 circulatory systems that are part of the cardiovascular system, okay? So they are not interchangeable terms, is my point, right? The lymphatic system contains the immune system. The cardiovascular system contains the circulatory systems, okay? So the immune system is what enables your body to either avoid infection altogether, or if you get infected, to limit the effect of that infection, okay? We have 2 different kinds of infection. We have two different kinds of immunity. We have innate immunity and we have adaptive immunity. And of the things that you need to understand in order to understand the rest of this process, this is a big one, okay? Understanding the difference between innate and adaptive is huge. Can anybody tell me their understanding of what innate immunity means? So it's immunity that you are born with, okay? When you come out of the womb, not the wound, although technically, when you come out of the womb, you are immediately protected by your innate defenses, okay? This includes barriers, right? Your skin is an innate defense. Your mucous membranes and the production of mucus is an innate defense, okay? The important thing to understand about innate immunity is that it is generalized. It is not specific to a pathogen, okay? This is a general overall, we're going to try to defend you kind of a situation, right? Inflammation is a response from the innate immune system, okay? It doesn't have to have a specific pathogen to be triggered. Inflammation happens regardless, all the time, okay? Adaptive immunity. Somebody tell me what adaptive, what's different about adaptive immunity? You acquire it over your lifetime. How? By getting, do you have to actually get sick? Getting exposed. Is that what you were going to say? Yeah. Getting exposed, right? Being exposed to a pathogen, a specific pathogen, will trigger a specific adaptive immune response. Okay, so you see the difference between adaptive and innate here. Innate, very generalized, non-specific. Adaptive immunity is a learned response to a specific pathogen. Okay, make sense? Questions? No? Okay, moving on. All right. So when we talk about innate defenses, a lot of the cell types that we've talked about up to this point are innate defenses, okay? Your neutrophils, your macrophages, the monocytes and basophils, all of those white blood cells that we talked about, most of those are innate defenses, right? They don't have to be trained to target something. They naturally target whatever it is, okay? Mast cells, dendritic cells, and eosinophils, all of those. And then we talked briefly about natural killer cells the other day. I think I asked you guys a question about these because they were in the blood chapter too. Okay. I'm like, why did I ask a question about these when we were talking about blood? Because they were in that chapter too. So natural killer cells are particularly useful form of innate immunity. Okay. So here's the thing. When we talked about meiosis way back in A&P1, we also talked, sorry, not meiosis, mitosis. We also talked about cancer, right? And we defined cancer as a very broad, in a very broad way. How did we define cancer? Uncontrolled cell division, okay? Any uncontrolled cell division can generate a tumor, okay? This happens all the time in your bodies, all the time, right? Exposure to ultraviolet light can trigger a mutation that leads to uncontrolled cell division. There's any number of things that can happen that can lead to this result. The natural killer cells in your body are designed from birth to identify cells that have gotten out of control, cells that are not behaving normally. And most of the time, when your natural killer cells detect those abnormal cells, they will destroy them, and therefore you do not develop cancer. Sometimes they get by, right? And that's when things happen and you have to go see the doctor and they do their thing. But that is what natural killer cells are for, right? This is from birth, they don't have to learn, oh, that's a cancer cell, or this is a particular kind of cancer cell. It's, you are not normal, you are not following the rules, I'm gonna eat you. That's the way it works, okay? I mean, that certainly encourages following the rules. Certainly would follow the rules if I was gonna get eaten if I got caught not following them. Okay, any questions about any of that? You should know what the cells are. You should have a general idea of what they do. Okay, you should know that they are innate, meaning that they are automatic, generalized defenses. Okay, we talked about the fact that inflammation is an innate defense, okay? So there are words over there on the left, and you should learn them and understand the definitions, but I want to look at this. So this figure can look a little scary, okay? But if we follow the numbers and pay attention to what's happening, it becomes a little easier to follow, right? So what kind of injury or damage do I have here? I got a splinter, okay? Anybody here ever gotten a splinter? Is the splinter itself the problem? What's the problem? It's not the inflammation. It's not the damage to the tissues. It's the bacteria on the wood. You have inserted bacteria past your initial barrier defense, right? Bacteria that would not normally be inside has now gotten inside because it rode in on the splinter, okay? That is what these little green dudes are. Oh, that's not a good color for that. Let's try green. That's these little guys up here. Is that better? Yeah. Okay. 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 All right. So we have brought in bacteria. The bacteria being in there is actually going to increase the amount of damage done to the tissue. Okay. Because of that, cells in the area are going to release chemical mediators. And those chemical mediators are going to go out into the body, and they're going to encourage chemotaxis. What is chemotaxis? When the cells go towards the signal, right? The chemical signal goes out and says, hey guys, come this way, right? Little Pied Piper. Anybody know that reference? I just use a reference nobody knows. Okay. Happens when you get to my age. Okay, so we have chemotaxis, and the chemotaxis is going to do a couple of things. It's going to increase vascular permeability. What does that mean? Vascular refers to what? The blood vessels, okay? They're going to become more permeable, meaning things are able to move through them easier, right? So the reason for that is that these particular vessels are capillaries, okay? And specifically, they are something called fenestrated capillaries, okay? What that means is that along the edge of the capillary, there are little spaces. Okay. As the inflammation happens, one of the things that comes along with inflammation is a chemical that causes vasodilation. What does vasodilation do? Makes the blood vessel bigger, right? So we go from a small capillary to a bigger capillary. When I go from a small capillary to a bigger capillary, shrink my eraser down here, my spaces get bigger. So now I'm more permeable, right? These fenestrations, these gaps get bigger to allow larger cells to be able to squeeze through them. Okay, so that is the increase in vascular permeability. The increase in vascular permeability is also going to cause edema. What is edema? Swelling. Okay, this is things get red, they get swollen. You ever notice you get that little bump, even if you can't feel the splinter itself, you feel the little bump of skin over top of it, right? That's the swelling, the inflammation, and the increased blood flow that we just talked about is what's going to make it red. Okay. Now, along with this chemotaxis, we get an influx of white blood cells, specifically neutrophils. I think I may have spelled that wrong. Neutrophils. Okay, and the neutrophils squeeze out of the blood vessel and they come up and they basically attack and phagocytize the bacteria. What does that mean? They eat it, right? The immune system is all about eating things. Okay. Now, once the bacteria get destroyed and broken down, and they're gone, the tissue will start to repair itself. If there are any bacteria left, then more chemical mediators will be sent out, increasing the chemotaxis, calling in more cells to actually try to finish the job. Make sense? Kind of a cool process, huh? I always think it's interesting. Okay, anybody have any questions about that? No? Okay. A couple of things I want to point out. When all of those dead pathogens happen, y'all ever notice that when you get a splinter, that sometimes you'll get like a white pus underneath of the skin? That is the dead pathogens and debris from the dead tissues that's been left behind after the neutrophils have eaten everything. That's what that is. Yes. Yes. White blood cells, dead pathogens, cell debris, all of that stuff is what makes that pus that gets left behind. Fever, systemic inflammatory response triggered by substances released by macrophages. So when you get sick, it's not specifically the bacteria that causes your fever. It's the immune system raising the temperature in an attempt to kill off whatever it can kill off. Okay, so a low-grade fever is not a bad thing when you're fighting off a virus. That's why I didn't do anything about mine. I had 101 degree fever for like 2.5 days, day and a half. It's miserable. It sucks. But it's my body doing its job, fighting off whatever virus it was or bacteria. Septic shock happens when the inflammatory response gets out of control. Okay, if the inflammation just sort of goes wild and it becomes too bad, septic shock will occur. Chronic inflammation, which most of us suffer from, can actually threaten human health. Chronic inflammation causes fatigue and pain, joint pain, muscular pain, swelling. It's just a miserable existence. Yeah, right there with you. A prime example right here. I live with chronic inflammation. Yes. It can, yes. So any sort of bacteria entering into the skin can cause this inflammation response. And so, yes, if bacteria get into your skin and you end up with a pimple, that is your body fighting off that bacteria and leaving behind all of the dead, decaying stuff. Lovely, right? Lovely, right? All right. Any other questions? No? Okay. All right. Moving on to adaptive immunity. Now, you'll notice a theme here. Okay. The B cells and the T cells. So I had a question a minute ago about why one is named B cells and why the other one is named T cells. And it's because they are named based on where they mature. Okay. The B cells mature in the bone marrow. The T cells mature in the thymus. Okay. So it's not where they're developed, it's not where they're made, it's where they mature. Okay. basically think of it as their training site where they get trained. Okay. The T cells need that more than the B cells. Now, there are some groupings I want you to pay attention to here. So, what's happening? Okay. So B cells, plasma cells, and memory B cells, they all go together. Okay. They all go together. A B cell actually produces or becomes a plasma cell, and the plasma cell then makes antibodies. Okay. So when you think about B cells and antibodies, it's not the B cell itself that's going out and defending the body. It's the B cell giving rise to or producing a plasma cell. That plasma cell then makes an antibody specific to an antigen, and the antibody goes out and does the job. So I like to sort of imagine the B cells more like generals, right? They sit in their office and they say, hey, you go take care of that problem, right? They don't really get their hands dirty, okay? We will talk about memory cells in a minute, but just understand that a memory B cell is a form of a B cell, okay? It's basically a B cell that is smarter. And I'll explain why that is in a minute. Now, we have the same thing here. T cells, cytotoxic T cells, helper T cells, memory T cells, dendritic cells get involved in here as well, but we're not going to worry about them as much, okay? Now, I will say this. Helper T cells can involve themselves in the B cell process. They can do either, okay? The T cells that mature in the thymus will eventually become either a cytotoxic T cell, a helper T cell, or a memory T cell. Okay? And we'll talk about the differences between them in a few minutes. Dendritic cells are one of a number of different cell types that we refer to as antigen presenting cells. Okay, there are others of those. We'll talk about them in a minute. Anybody have any questions about those? This is all just terminology and lingo. Okay, we haven't quite gotten to the how does this thing work process yet. Is with me? Mm-hmm. They are antigen presenting cells. Yes. We'll talk about what that means in a minute. Okay. Moving on. All right, so this should be easy. We talked about this, and I told you, I warned you it was going to come back. What is an antigen? They're markers on the cell, right? Antigens are markers on a cell, okay? In terms of the immune system, an antigen is going to mark a pathogen. Okay, so we talked about antigens in blood typing, right? And remember, we talked about the antibody that attacks the opposite of what's on the red blood cell. That's why, right? It perceives that antigen as an enemy. Okay, antigens to the immune system are the enemy. Okay, depending on the antigen and depending on the antigen receptor, we are either going to trigger a response from the B cells, which will generate an antibody, or a response from the T cells, which will cause what we refer to as a cell-mediated response. Okay. Now, there are millions of different antigen receptors. Okay. All kinds of different antigen receptors. And antigen receptors are specific to antigens. Okay. So each individual antigen, remember, we're talking about adaptive, right? When we talk about adaptive immunity, we're talking about specificity. So each individual antigen has a specific antigen receptor that it is going to attach to. And that is how we're going to trigger this response. Does that make sense? Okay. Now, on an individual cell, you can have lots of antigen receptors, but they are all going to be for the same antigen. Does that make sense? So an individual B cell, if you look at these two cells, right? So on this B cell, my antigen receptor is specifically shaped like that little V, right? All of the antigen receptors on that particular cell are that same type, right? The same with the T cell. If you look at the antigen receptors on that T cell, all of the antigen receptors on that specific cell are the same. Now, a different T cell could have a different antigen receptor. Okay? Does that make sense? Different pathogen, right? So T cells and B cells are going to be trained to detect very specific pathogens. And how they are trained is by developing these receptors. So each pathogen that the body is exposed to creates a set of B cells or T cells with receptors specific to that pathogen. So you could have B cells specific to, I don't know if those antibodies or not. I think that's T cells. T cells specific to chickenpox, or T cells specific to mumps. Right. Each T cell only defends against one pathogen. They'll have multiple receptors for that one pathogen, but it's only one pathogen. Okay. These are very much singular focused cells, right? They do one thing, they do only one thing, and they do it as well as they can. Okay. Yes, there can be. Yes. And we'll talk about how that works in a minute. Okay. You guys following me so far? This is the most confusing system in your body. So I'm trying to make sure that what we're doing is clear. Is everybody clear so far? Okay, we're not even going to get into the really complicated stuff. This is the easy part. Okay. Now, up at the top, okay? We're just looking at the top. We're ignoring everything on the bottom because it's got that scary word that I'm sure all of you freaked out about, that major histocompatibility complex. We're going to ignore those for a minute. Up at the top, we have B cells, okay? Now, when a B cell has been trained to detect a pathogen and it encounters that pathogen, that is going to cause the B cell to produce a plasma cell. Okay? That plasma cell is specific in terms of what kind of antibody it will produce. Right? It will secrete an antibody. Another word for antibody is immunoglobulin, and you'll see them abbreviated as IG very frequently. There are several different kinds of immunoglobulins. We are not going into those details. So if you choose to read that in the textbook and make your brain explode, that's on you. We are not going down that rabbit hole. Okay, it is quite a rabbit hole. If you're really fascinated by them, take immunology. Dr. Buck loves to talk about the globulins. Feel free. Okay, the plasma cell is a soluble form of the B cell that will make. Sorry, the antibody is a soluble form of a protein. The antibody will then go out and attach itself to the cell that has the antigen. Okay, and they'll surround it and they'll bind it up. Lots of little antibodies all surrounded him and holding him prisoner so that then a macrophage can come along and eat them. Make sense? You guys with me? No? Which part? How far back do I need to go? All the way back? Okay, let's see. All right, so we have a cell with our antigen, our receptor, and our antigen. Okay, that is going to cause the B cell to make plasma cells, which we're going to make blue. These are plasma cells. Okay, the plasma cells are going to then make antibodies. Okay, the antibodies are then going to find the pathogen, okay, and surround it. So let's say I've got my cell over here and I've got my pathogen. Well, this is my pathogen cell. Okay, I'm going to have all of these little guys. They're going to come over here and they're going to attach themselves like a little army. Okay, and they're going to surround that bacteria. Okay, so one of the things that's important for you to understand, antibiotics are a bacterial defense. Okay, antibodies are associated with antibiotics. Okay, so you're primarily looking at bacterial defense here, right? So once these antibodies have surrounded my little pathogen, then my macrophage is going to come along and eat him, break him down.
So those antibodies are immunoglobulin, or is it just...
They're made of immunoglobulin. Yes. Yes. Does this make sense? I love the fact that my horrific drawings actually make sense to you guys. Did you guys follow that better? Yes. Okay, so I have my pathogen surrounded by my little green antibodies, right? I have Pac-Man over here, right? He's going to come along and he is going to engulf and eat that. And then inside of it, it's going to break it down into basically waste product pieces. And it destroy the cell and pathogen until he's just all waste product. What's the pathogen? The macrophage. Macrophage. The yellow thing is the macrophage. Pac-Man is the macrophage. Phagocytic cells. Macrophage, big cell that eats things. Yeah. Yeah. That's exactly what they are. They're like a red flag to a bull. Make sense? Yeah. That's a B cell. That's a B cell that has an antigen attached to it. So this B cell has been trained due to exposure, and we'll talk about how exposure works in a minute. But it's been trained due to exposure to know how to fight this particular pathogen. and to know which antibodies are going to be used to fight this pathogen. Okay, so as soon as a fragment of the pathogen is detected by the B cell, then the B cell will make the plasma cells. The plasma cells will make the antibodies. The antibodies will then target the actual pathogen, surround him, and call in the macrophage. Yes. They're made of immunoglobulins. So an antibody is made of immunoglobulins, sort of like an omelet is made of eggs. Food references do it every time. Okay, make sense? Okay. Let's talk about the scary guy. Major histocompatibility complex molecules, MHC molecules. You'll often hear me refer to them as MHC markers. Okay. These are host proteins that will display an antigen fragment. Now, here's the deal. In order for an antigen fragment to be displayed, the pathogen has to have been inside of the cell that carries the MHC marker. Okay, so let's talk about this, right? So B cells respond to extracellular or intracellular pathogens? Extracellular, right? They're looking for a fragment that broke off that's floating around and it's going to encounter that receptor. T cells are looking at intracellular pathogens, something that is inside of the cell that has been broken down, and then the little tiny fragment, so you can see it happening here, okay? The cell, and we're going to talk about what cell this is. This is not the T cell specifically. This is a host cell that has this marker. So this is our pathogen, and our pathogen, oops. Let's go with white. Our pathogen is going to get taken into the cell. Okay. When it gets taken into the cell, it gets broken down. So when we take it in, right, we take the pathogen in, and the host cell, let me give you another name here, antigen presenting cell. Okay, so host cell equals antigen presenting cell. Okay, think of T cells as like the royal snobs, right? In order for them to do anything, somebody has to present the antigen to them to let them know there's a problem. Okay, so that's our host cell that we're talking about. Our antigen presenting cell has taken in this pathogen, identified the pathogen, has created the appropriate MHC marker, moved it to the outside of the cell, broken down my pathogen into little fragments, and then I'm going to move those little fragments out to the outside of that marker. Okay, so all of this is coming from inside of the cell out. Okay, once it's out, it's then going to be presented to our T cell, who will then attach to the antigen, which is still attached to the MHC complex or marker, which is still attached to the host cell. Everybody stays attached. Does that make sense? You see how this is very different from the way that the B cell works, right? The B cell never gets its hands dirty, right? The hint of a pathogen has come by and the B cell's like, ooh, dirty. plasma cell, make an antibody and send them out to go find that. I don't want to touch it. I just want you to go do the work, right? The T cell, a little bougie, right? Requires it to be presented, but once it's presented, it's like, okay, I've got it. I'll take care of it. Make sense? I told you guys I have cartoons in my head, right? Like all the T cells have little crowns. And the presenting cells, the antigen presenting cells in my head are like little ring bearers at weddings with little antigens on pillows, walking around showing them to people. Yeah, that's what's in my head. Can't help it. It's the way my brain works. Okay, so the T cell will bind to the antigen fragment, and then the T cell will actively participate in destroying the cell. Okay, so does everybody understand the difference here? Because we're going to dive a little deeper. Okay, now this is true of both B cells and T cells, although it's a little different in how they work. Okay, both B cells and T cells undergo something called proliferation. Now, we've used this term before. What does proliferation mean? Does it mean to proliferate? Nope. Bunnies do it really well. Works every time. Proliferation, to make more. I used to use nuclear proliferation because it was a thing in the 80s, but apparently not anymore. So bunnies are very proliferative breeders, right? They make more very rapidly, okay? So proliferation of B cells and T cells means that the B cells and T cells are going to make more of themselves, okay? They are going to clone themselves, okay? It is different from mitosis. It's a similar process, but it's not technically mitosis, right? Remember, mitosis is specifically the division of the nucleus and then the cytoplasm. We're not dealing with that here, right? Not all of these cells even have nuclei, okay? So what we're going to end up with are clones of the cells, but we don't want to clone a cell that doesn't know anything, right? Like if I were to clone myself, I would want to clone myself now, not when I was 18, right? I definitely need more of me now that I know some things, because I was dumb at 18. Thank God there was no internet back then. There was, but it was like AOL and dial-up, so it's not something you guys ever experienced. So they want to make clones after they've been exposed to a pathogen. They want to clone themselves after they've figured out what they need to do to defend against that pathogen. Okay? So once they get activated, once either the B cell has detected a pathogen or the T cell has been presented to the pathogen and identified it, then they will undergo clonal selection. Okay? They will duplicate themselves like a little photocopier. Okay? Now they're going to make two different kinds of copies. Okay? They're going to make effector cells and they're going to make memory cells. What does an effector do? It causes an effect, right? It actually does something, right? Just like we talked about skeletal muscles being effectors and actually causing movement, right? So the effector cells are the immediate response. Okay, we've detected the pathogen is a problem, we know how to kill it. Where in the case of B cells, we're going to make plasma cells. The plasma cells are the effector cells because they generate the antibodies that defend, right? The T cells, the activated T cells, are actually the effector cells on that side. Okay. So they're actually going to go out and do something. They're not going to last very long, right? They're not going to stick around very long. They're going to cause a response and they're going to die off and go away. Okay. The memory cells are longer lived. The memory cells stick around just in case you happen to run into that same pathogen again. Because now you already know how to defend against it and you can respond faster. Does that make sense? Yes. This is all adaptive immunity, right? Remember, in order to activate, they have to identify a specific pathogen and figure out how to respond to it, right? So the whole thing is adaptive. The difference is one of them is adaptive and responds immediately. That's the effector cell. The other is adaptive in that it has learned, but we're sort of saving it as in the reserves. It's like our reserve soldiers, right? They're going to hang out just in case. It's the reason why you only get chickenpox once, right? You get chickenpox once because you were exposed to chickenpox, or not anymore. Now you don't really get it. Now you get exposed to a vaccine, right? Does that mean you're never exposed to chickenpox again in the world? No, you're still exposed. You still can have the virus actually invade your body, but your body knows how to kill it. COVID vaccines also do the same kind of thing. Now, long-lived does not mean forever, right? For some viruses, it does. For some viruses, we have memory cells that last our entire lives, right? Others require updates. Yep, that's what booster shots are for. That's why every 10 years you have to get a tetanus shot. Okay, because it lasts, but it doesn't last forever. Does that make sense? Yeah, that's your memory cells. Okay. How are we feeling? How many of you looked at this graph? Chickens. Okay, so this graph explains everything I just said to you, only it does it visually, and it's a beautiful graph. All you have to do is really look at it. So what are we looking at here? Tell me what this graph is, just generally what this graph is showing. The change in antibody concentrations in arbitrary units over days, over time in days. Okay. Now, if the antibody concentration goes up, what does that tell me is happening? You're having some sort of an immune response, right? You've encountered a pathogen, you are developing some kind of response, right? Is this a T cell or a B cell response? B cell, why? Because it's antibodies, right? B cells make plasma cells, which make antibodies. Okay, T cells don't have anything to do with antibodies. Okay, so this is a B cell response. Okay, now we have two different pathogens in this graph. We have pathogen A and we have pathogen B, which are identified in here based on their antigens that are being detected by the B cells. Okay, so we have antigen A and antigen B. Now, on day 3-ish, you get exposed to antigen A. Right? How long does it take for your body to start responding to that antigen? 3 or 4 days, right? There's a time lag. You see this here? Give me this. Right here, from the time of exposure to the initial time of response, there's a couple of days in there, right? That is your body identifying the pathogen and trying to figure out what it needs to do to fight it. Okay, this is the learning, this is the training. Okay. Now, once we've learned and we've trained, now our B cells are going to clone themselves, right? And they're going to make both effector cells and memory cells. Okay. The effector cells are going to do what? They're going to what? They're going to act right away. And how are they going to do that? They're going to create antibodies. So we see our antibodies increase. Okay. Remember, they're short-lived. They don't stick around very long. They just stick around long enough to do their job, right? So as they do their job, the antibody goes down. Now, what do you notice happens? I don't think I can get any bigger with it. What do you notice? It dips, but it doesn't go to zero, right? I have space. I don't go to zero. Why don't I go to zero? Yes, the memory cells. The memory cells are down here, right? And they're just hanging out. Okay, now, right around day 28, because somebody decided to come to class sick, you get exposed to antigen A again. And at the same time, because somebody else showed up sick, you also get exposed to antigen B. Okay, so you've now been exposed to both at the same time. Let's talk about first what happens with your antigen B response. What does antigen B response look like? It lags again, right? I'm going to switch to a blue line because that makes sense to me. So I've got another lag before I start. Why? Because it's learning again. It's a new antigen. It doesn't know what to do with this one. It has to figure it out. So it's going to have a primary response to antigen B. It's going to take longer to figure it out. It's going to generate antibodies, but it's going to generate a relatively low number, right? They're going to go away pretty quickly, but they're not going to go away to zero because I still have memory cells. I now have memory cells for antigen B. Now look at what happened to antigen A after my second exposure. Was there a lag in response? No. What do you notice about the difference in the initial response on day 6 versus the secondary response on day 29? Much faster, it's much higher, right? The slope is almost non-existent. It goes straight up. And when I drop, what do you notice? It doesn't drop as low. I have even more memory cells left. Yep. Now, is it possible that this will eventually drop to zero? Yes, but these are going to last a lot longer. Second exposure is going to last a lot longer. So this is the difference between a primary exposure and a secondary exposure, or a primary immune response and a secondary immune response. Each new pathogen starts out with a primary immune response. Make sense? Okay, on Monday, we will finish chapter 22. Yes. Are we going to look at what? Oh, you know what? I forgot you guys finished the test. We will do that on Monday. Yes. Completely forgot you guys finished the test. Anything come up overnight? Okay. So we talked about the B cells and their sort of clonal functions. Now we're going to talk about the difference between a humoral immune response and a cell-mediated immune response. Okay. Now the word humoral should sound familiar. right? Talked about humoral hormone control. In this case, when we say humoral, you need to immediately think about B cells. Okay, humoral immune response requires antibodies to neutralize or eliminate any sort of toxins or pathogens in the blood and in the lymph. Okay. Cell-mediated immune responses are T cell related. Okay, where the specialized T cells, cytotoxic T cells generally, will find and kill host cells that contain pathogens. Okay, humoral deals with extracellular pathogens, things that are not in the cell. Okay, they're in the blood. Cell-mediated responses deal with intracellular pathogens, things that are in the cell. T cell is cell-mediated, B cell humoral. Okay, there's a figure we're going to look at the end that will sort of bring all of this together. Now, there is one T cell that we talked about that sort of walks the line between the two. Okay, the helper T cell is capable of triggering either a humoral response or a cell-mediated response, depending on the kind of pathogen that it has encountered. Okay. In order for this to happen, the helper T cell has to bind to the antigen. What does a helper T cell require in order to bind to an antigen? The receptor has to be on the T cell. An antigen presenting cell, is that what you said? APC. I'm like, wait, I don't know that. I don't know that abbreviation. What was that? Antigen presenting cell, right? Remember, T cells are bougie. They need it. They need to be presented with their antigens. They're not going to go looking for them, right? So the antigen presenting cell has to display the antigen for the helper T cell. The helper T cell will then evaluate what kind of pathogen it's dealing with. Is it an intracellular or an extracellular? Do I need to alert the B cells or do I need to alert the T cells? Does that make sense? Does that make sense? Y'all.
Helper T cells will alert either B cells or other T cells.
Yes. 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 So antigen presenting cells will have one of two kinds of major histocompatibility complex molecules on them. Okay. They will have class 1 or they will have class 2. Or sometimes they might have the ability to make both. And we'll talk about that. Okay. Now, I used to do this in a big long process where I would draw things out, but I made my life easy and I created a table. Actually, we're gonna hop over here because this is the easier way to understand it. So the information here is related to the information here. This stuff is easier to understand over here. So major histocompatibility complexes come in two forms. There is MHC1 and there's MHC2, right? Now, there are similarities between them. All of them are found on the surface of molecules, okay? All of them will trigger some form of an immune response. Both MHC1 and MHC2 molecules on the antigen presenting cells will present to T cells, but they present to different kinds of T cells, okay? And interestingly enough, both MHC1 and MHC2 molecules on antigen presenting cells can cause rejection in transplant recipients. This is one of the reasons why transplant recipients are given immunosuppressants, because the body is constantly registering that transplanted organ as something that doesn't belong there. And so the antigen presenting cells keep trying to present it. And so we have to suppress that urge, okay? Now, here's the difference. MHC1 antigens, MHC1 molecules, okay, are found on nucleated cells. Name a non-nucleated cell. Not a whole lot of them, right? There are a few, there are not a lot of them. What that means is MHC1 molecules can be found on virtually any tissue in the body, any cell type in the body. If it has a nucleus, it has the ability to produce an MHC1 molecule. Okay? They are very common. MHC1 molecules are found on cells that are presenting an antigen to a cytotoxic T cell. What kind of antigen do I have to be dealing with in this case? Cytotoxic T cell. Extracellular or intracellular? Intracellular, okay? They're going to respond to intracellular pathogens, right? So basically what's going to happen is they're going to take in that cell, that pathogen, the antigen presenting cell is going to take in that pathogen, or will have that pathogen. It will break that pathogen down into fragments and then put up the flag, right? Say, hey, I've got an intracellular pathogen here. Come and help me, okay? What they don't know as they put up the flag is that they have now put themselves in the dead zone. Okay, because that cytotoxic T cell is going to come in and help them by destroying it. Okay, so friendly fire and all that. So go back to our normal military process. So that's MHC1, right? I'm a nucleated cell in the body. I have an intracellular pathogen. I have fragments of that pathogen. I'm going to make an MHC1 molecule. I'm going to display that antigen there and display it for a cytotoxic T cell to come and attach and kill. Okay. MHC2 molecules are also found on antigen presenting cells, but these are very specific cells. B cells, macrocytes, monocytes, dendritic cells. Now, do any of those cells have nuclei? They do. Which means what?
It can also be activated by the MHC.
Yes, they can also make MHC1. So an antigen presenting cell that is a B cell, a macrocyte, a monocyte, or a dendritic cell can respond to both intracellular or extracellular pathogens. Okay, this is why if I have an MHC2 molecule on an antigen presenting cell, it's going to get presented to a helper T cell. Because then that helper T cell will evaluate, okay, is this an intracellular pathogen that I'm working with? In which case I need to alert the cytotoxic T cells. Or is this an extracellular pathogen that I'm working with? In which case I need to alert the B cells. Okay, so MHC2 can be found on the same kinds of cells that some MHC1 are found on. Okay, now I want to be very clear about this. Just like when we were talking about the antigen receptors that are present on the B cells and the T cells, once an antigen presenting cell has committed to an MHC marker, that's the only MHC marker it's going to make, right? So if I'm a B cell and I'm hanging out with my other B cell buddies and I come in contact with an extracellular pathogen and I generate an MHC2 molecule on the outside of my cell, I cannot then also make an MHC1. But my buddy over there, my other B cell buddy over there, can make an MHC1 if they encounter an intracellular pathogen. Does that make sense?
So the cytotoxic T cells, they're the only ones that cannot respond to MHC2.
So MHC2 gets presented to a helper T cell, and the helper T cell can then activate a cytotoxic T cell. It's just an indirect pathway. Okay, so MHC1 is presented directly to the cytotoxic T cell. MHC2 will get there, it's just got to go the indirect route.
So the B cells, macrocytes, all those can respond directly to the MHC1 and MHC2? Is that what you're
Yes.
saying? Okay,
Yes.
okay. So
Yes.
the only one that is indirect, I guess, would be the cytotoxic T cell, as far as if it was.
So here's where you're getting, I think here's where you're getting confused. B cells are also antigen presenting cells. Okay, so B cells can present to a helper T cell, but as you will see, when that B cell gets engaged with that antigen, it will also go ahead and do its B cell thing. Okay, we're gonna look at a table in a minute that I think will help with this. Okay, and actually we may. Yeah, I'm gonna pop forward to this table because this table usually solves the confusion. Okay, so this table looks a little, this figure looks a little scary sometimes to students. There's a lot of arrows, there's a lot of stuff happening, okay? One of the things that you need to realize is that there are two separate processes happening right down the middle, okay? Over here, I have cell mediated. If I have cell mediated, then I'm going to be dealing with what? T cells. What specific T cells? Cytotoxic T cells. Intracellular or extracellular? Intracellular. Okay, so I'm looking for intracellular pathogens. Cancer is also something that gets dealt with over here, okay? On the other side, I have my humoral. Okay, so over here, I'm dealing with what? B cells. Okay. Intra or extra? Extracellular. Okay. What about MHC complexes? MHC2 is going to be where? Let's do the easy one. What about MHC1? MHC1 presents directly to the cytotoxic T cells. So this is definitely MHC1, right? Can I have MHC1 on the other side? No. No. MHC1 only presents directly to cytotoxic T cells. MHC1.
But it can't, but it's on nucleate itself.
But is it an MHC1 or an MHC2? So you get two different molecules. Not asking about the cells.
Oh.
I'm asking about the complex, right? The flagpole.
Oh, okay.
Okay, I'm talking about the flagpole.
Okay.
Okay. That seems to be a really good reference. I'm hanging on to that reference. All right, so my MHC1 flagpole only exists in cell-mediated immune response. It only responds to intracellular pathogens. Okay, now MHC2 responds to what? It can, right? So MHC2 is going to present to this guy in the middle, right? So it could be over here, or it could be over here, right? Because it goes both directions. Just depends on what kind of pathogen that MHC2 marker is dealing with. Does that make sense? Yeah. Okay, so I'm going to erase some of this because I get crazy drawn over this thing and then it gets to be a mess. All right, so what do we want to talk about first? Cell mediated or humoral? Okay, so like always, when we're reading a figure, the first thing we need to look at is what? What the arrows mean, right? What are the arrows telling me? So I have two different kinds of arrows. I have the black arrows with the little green pluses. That means that we are stimulating something. We're causing something to happen, okay? And then the brown arrows mean we're giving rise to. What does that mean? It's not really amplification, although you've got this right general idea. It's not really chemotaxis. It's more, it's a what?
Like a specialization thing, like one thing will turn into like the other thing.
Yes, yes. Giving rise to means that I'm going to become, right? So a cytotoxic T cell that has been stimulated is going to become an activated cytotoxic T cell. Does that help? Okay, so we have our antigen arriving, our first exposure of our antigen here, okay? And that antigen is going to be taken up directly by an antigen presenting cell. And that antigen presenting cell is going to do one of two things. What are those two things? Could stimulate a helper T cell if we have a MHC2 will send us to the helper T cell. If we have an MHC1, we go straight to the cytotoxic T cell, okay? So this is MHC1, this is MHC2. Okay, so we've got these two different pathways. If I have an MHC2 molecule, then I'm going to go to the helper T cell. If I have an MHC1, I'm going to go straight to the cytotoxic T cell. Okay, now if I'm using an MHC1 complex molecule, I go straight to that cytotoxic T cell, and what happens? Right, so the cytotoxic T cell is going to give rise to or become activated cytotoxic T cells. And through that clonal replication, that clonal selection that we talked about, this cytotoxic T cell that's now been exposed and trained, right, is going to develop a series of memory cytotoxic T cells. We're going to get both of those things. Okay. That active cytotoxic T cell is then going to go out and do its job. Okay, sending in the green berets. All right, now, if on the other hand it was an MHC2 molecule, what is going to happen then? So that helper T cell is going to stimulate the cytotoxic T cell because it was an intracellular pathogen. Okay. And we're going to go through that whole process, right? We're going to make memory cells. We're going to do all of those things. But we're also going to generate what? memory helper T cells. So we're going to have helper T cells that remember that particular pathogen, so that if we get exposed a second time, we can activate or stimulate the memory helper T cells, which are going to stimulate the memory cytotoxic T cells. And because it's an MHC, or because it's an intracellular pathogen, that memory helper T cell can also, that second exposure can actually directly stimulate those memory helper T cells, those memory cytotoxic T cells. In which case, all of those cells are going to go over here and become active, right? And we're going to generate some more of these, and we're going to make some more memory cells, and we're making all of these new cells and doing all of this stuff, right? Remember that second exposure is going to get us a faster, stronger response, right? Does this make sense? Is this easier to understand? I love this figure. I've had many students say, if you can understand this figure and really clearly understand this figure, you can basically answer most of the immune system questions on the exam. Okay, so let's go the other way. Humoral, my antigen exposure. So my antigen shows up. What am I dealing with? What's happening? It's going to stimulate the B cell, right? It's going to directly stimulate the B cell. All right. What else is it going to do? It's going to be taken in by the antigen presenting cell, which is displaying what kind of marker? MHC2. And that's going to go to stimulate the helper T cell. The helper T cell is also going to stimulate B cells, right? And all of those well-stimulated B cells are going to do two things. They're going to give rise to memory B cells that remember that extracellular pathogen, and they're going to give rise to the effector cells, the plasma cells, which are then going to secrete antibodies and go out and fight those extracellular pathogens. If I have a second exposure, same deal, right? I'm going to stimulate my memory helper T cells. They're going to stimulate my memory B cells. My memory B cells are going to create more plasma cells. We're going to generate more antibodies, and those antibodies are going to go out and fight. Does that help? Much easier, right? Once you kind of can put a direction on it, the MHC classes they're not quite as difficult, right? I know they're scary when you first start learning about them, and they can get really complex. It can get really, like, you can deep dive into these guys, but we're not going to do that. All right. Ooh, what happened? No, I don't want to do that. I don't want to do that. I want to do this. Okay. So let me back up just to make sure we haven't missed anything here, because there's a couple of things. Let's see. Did we talk about this? Antigen receptors. Helper T cell is activated. Yep, we covered all of this. Zip forward here. It's important to understand that the antibodies, when they're produced, what they're going to do is they're going to go out and they're going to attach to the pathogen. And there are several of them. They're going to gang up on him, right? They're going to all surround him and gang on him and, pin him to the floor. And then the other little ones are going to bring more of his little buddies over, and they're going to attach them all together. And then a macrophage is going to come and eat the whole mess. Okay. It's a cartoon in my head. It is. It's a great little cartoon in my head. I'm telling you. I could draw well enough, I would make a fortune. I've heard, I have heard good things about it. I have not watched it. I've not seen it, but I have heard good things about it. So, yeah. I have heard good things about it, yes. Okay, so the activated cytotoxic T cell, like I said, you know, we raise our little flag and the cytotoxic T cell comes over in the guise of helping and destroys us. Right? Okay. I know it's very sad. All right. Moving on to active and passive immunity. Okay, so active and passive immunity each come in two separate forms. Okay, active immunity means your body has to actively produce the defense. Okay, passive means that you are given the defense. Now, here's where this gets confusing, right? Because artificial active immunity is, a lot of people think, oh, we're giving you the defense. We're not actually, and I will explain to you why. So we have, I'm just gonna, oh, no, we're not gonna do that. Oh, that's what we're gonna do. All right, so we have active and we have passive. Okay. Now, active immunity comes in two forms. We have natural and we have artificial. Okay. Natural active immunity happens when I get the flu and my body produces antibodies. Okay. That's natural. I naturally got exposed out in the world and I produced the defense in my body on my own using my own energy expenses. I actively produced the defense to kill it. Okay. Artificial is when I am exposed to an inactive form of the virus via a vaccine. And the antigens in that vaccine, although they don't actually make me sick, I don't generate symptoms, they do stimulate the development of the antibodies in relationship to that antigen. So my body still has to produce the antibodies. This is why the fact that we have to produce the antibodies is why you can get sick after you've gotten the flu vaccine. It does not mean that the flu vaccine made you get sick. Unless you're getting the nasal version, which is the live virus, the flu vaccine is inactive. It's not going to make you sick. But if you are exposed to the virus naturally, between the time that you got your vaccine and your body actually created the antibodies, you are still susceptible, right? That primary response, that lag that we saw, that lag means you're still susceptible to the virus until you've started producing those antibodies. So this active process is your body having to produce antibodies. And so there is a period of time where you're still susceptible even after you get the vaccine. Okay, it's not like they shoot you with the vaccine and you walk out and you're like, bring it on, right? Your body's like, no, wait, I'm not there yet. Now, passive also comes in a natural and an artificial form. Natural passive immunity. There's a very short period of time in which we all have natural passive immunity, and it is directly after we are born. And it is transmitted. Some of it is transmitted directly from mom before you're born. Some of it is transmitted through breast milk. So if you breastfeed, you are increasing the natural passive immunity that your child has. If you don't, they still have some, because you give them that when they're developing inside of the body, but they don't have as much. Yes, because that is it's full of antibodies and immune stuff. So this is mom to baby. Okay, baby's body is not making antibodies. Baby's body is not learning how to defend itself in relationship to this. This is simply mom passing on fully formed functional antibodies. Okay. Artificial. This is the one that most people struggle with. So there's a couple of examples of this that are useful. The first is an anti-venom. Okay, or like a rabies vaccine. Okay. Anti-venoms and rabies vaccines, those things have to be given after exposure because they are designed to kill the active virus or pathogen in the body. Okay, so you get bit by a snake, you have that pathogen in your body, the anti-venom is given to you because that anti-venom will go and attack that venom.
Is that like,
Tetanus shots you get ahead of time, but they only last a period of time. So that memory that comes, so tetanus shots are actually an artificial active. You get the tetanus shot, your body develops antibodies, but the memory cells only last around 10 years before they start to get weak and you have to get a booster. Okay.
So how come some doctors, if it's been 5 years or more, if you, let's say, get a fish hook in your hand or your arm, give you a tetanus shot right then and there?
Because the fancy word efficacy, the benefit associated with that shot declines over time. Okay, it's the same thing with COVID, right? You get your COVID vaccine or you get exposed to COVID, right? And for the first 30 days, you're good, you can't get it again. But then you slowly become more and more susceptible over time, which is why they keep coming out with the boosters to try to increase that, to try to decrease that susceptibility. Okay, so the first couple of years after a tetanus shot, you're good, but it's better to be safe and give you the added boost than to risk you not having enough antibodies on your own, right? So your body still has to respond to that inactive virus by making more, but you're not likely to get tetanus from the inactive virus. Does that help?
It all depends on like how long your memory cells last, right?
Mhm.
And so it just depends on what kind of pathogen you encountered and like, because not all memory cells are like.
Not all memory cells are created equal.
Yeah.
They are not. They are not. It's why it's part of the reason why you have to get a flu vaccine every year.
Yeah.
Right? Some of it has to do with the length of time the memory cells last. Some of it has to do with the fact that the flu virus mutates so frequently. They can't cover everything even under the virus. Now, passive artificial. Do you guys remember back, not early in the pandemic, but sort of maybe a quarter to a third of the way in, when they started really starting to figure out how to deal with COVID, and they were asking people who had had significant cases of COVID to come in and donate their antibodies? That was for something called monoclonal antibody therapy. And basically what they were doing was they were filtering people's blood. They would centrifuge the blood, take the section of the blood that has the white blood cells in it out, right? And then they could transfuse those antibodies directly into patients who were having a hard time recovering on their own. So this was a passive artificial. We weren't giving them the antigen in order to encourage their body to make antibodies. We were giving them antibodies directly to help their body fight off the existing virus. Does that make sense? Okay, so there are a number of different artificial passive, they're just a little weird, right? Most of the examples that you're going to think of in relationship to your body's response to pathogens, in terms of vaccines, in terms of those sorts of things, most of that is going to be active artificial or active natural, right? Passive natural only happens once in your life. Passive artificial only happens under very specific circumstances. That makes sense? Usually, yes. Usually, yeah. Cytotoxic T cells, I mean, you can, I suppose you could transfer activated cytotoxic T cells, but I'm not really sure how that would work. Most of what you're dealing with is going to be some of the gene therapy. Some of the gene therapy might be considered like cancer gene therapies that they've come up with that target very specific genes might be considered a passive artificial. And that would be more related to the sort of intracellular side of the path. Yes. Yes. Whether it's an active virus or an inactive virus. If you get contact with the virus and then make the antibodies, then you are actively involved in the immune process.
Active artificial, basically the artificial part is they're given the antigens, that way we can produce antibodies.
Artificial basically means that you went to the doctor and got it, right? Like when I was young and they didn't have the chickenpox vaccine, it was all active, natural transmission, right? I'm gonna stick you into bed with your sister and hope that you naturally catch chickenpox, right? We're gonna have a play date with everybody in the neighborhood and try to get everybody to have chickenpox all at one time, which you would think would be a nightmare. But any parent who had more than one child only wanted to deal with chickenpox once. Get it done, get it out of the way. All right, does that make sense? Did this clear that up for you guys? Yeah, okay. All right. Allergies, whether they are like food allergies or hay fever, any of those things are hypersensitivity responses of the immune system to antigens that we call allergens. So I always like to point this out. Anybody in here take an antihistamine or an allergy pill, right? So that allergy pill does not prevent, it doesn't prevent the allergy. It just prevents the response. Yes, and it does it by blocking the receptor, right? So the antigen receptor gets blocked by the antihistamine, which means that that allergen, that pollen antigen, can't get to the receptor to trigger a response. Okay, so you're still allergic to pollen, you're just not feeling the response because your body can't, because the receptors are not available. Make sense? Okay. Acute allergic responses, we all know what anaphylaxis is, right? We all know that EpiPens are useful in short-term correction of anaphylaxis, right? You know, if you use an EpiPen, you still gotta go to the hospital because it's not gonna last forever. Okay. Anybody have any questions?
Why, even though like, so epinephrine, I know that they use it all the time in like ERs and stuff.
Mhm.
Why to purchase it for people that have allergies where it causes that? Why is it so expensive when you just get it?
I'm gonna sort of go a little political here. Big pharma. I mean, it's the same reason insulin is so expensive,
Yeah.
right? There's no reason for insulin to cost what insulin costs.
No.
No reason.
Needing insulin could literally kill you because of how expensive it is.
Right, but they don't care.
I say I put it every time my entire life.
It's the almighty dollar at work. So yeah, they charge what they can charge because it works. Now, to me, if I were a pharmaceutical company, I would make insulin and sell it at a lower price. Because
You know people are gonna need it.
everybody's gonna buy it from me.
Yeah.
I mean, Tylenol, when the generic brand of Tylenol came out, guess what most people buy? The generic brand, because it's cheaper. I don't understand why nobody has come up with a generic insulin. Not that complicated.
Yeah. The weird thing is if you look into the journals of Sir Frederick Banting, who created insulin,
Mhm.
he says he did not want to be the way it is today.
Oh yeah, it's big pharma. Yeah. Is that the idiot that bought the patent and then tried to crank it up to some ridiculous? Yeah. That man is lucky he didn't get assassinated on the side of the street somewhere. He is lucky. He's lucky somebody didn't just put him out of his misery.
I didn't know like big pharma was like, are like people within the stock market for manipulating the stocks of big pharma like that.
Oh yeah.
I didn't
Oh
like,
yeah.
I only like research.
Yeah, all that Wall Street craziness is why I don't mess with the stock market. I don't trust those
I'm
people.
like, I do know that it will cost money because like it needs like a purification process, and because of that purification process, like it's gonna be a whole bunch of like equal money.
Right, and that's fine.
Yeah.
Make a profit. I have no problem with making a profit. I have a problem when your CEO is making, $1 billion a day on a drug that costs, less than a penny to make a vial, right? I mean,
When people need it so badly.
yeah, I mean, my niece was paying for her diabetic dog $400 a month. She was paying for insulin for this dog. Yeah, we're a little extra when it comes to the animals. When I die, if I get reincarnated, I want to be reincarnated as one of my family's pets.
With allergens, you can get different allergic responses to the point where like some people vomit and get nauseated, where some people get.
So it depends on how your individual immune system reacts to each individual antigen. Some people have a much stronger reaction. I mean, peanut allergies, right? Peanut allergies can vary from, I don't have any and I can eat peanuts all day long, to sometimes if I eat too many peanuts, I get kind of an itchy feeling in my mouth, to if I breathe peanut dust that was in the air a week ago, I go into anaphylaxis, right? There are people who cannot be in a room where there were peanuts. I don't know how those people live life.
I don't know.
I
how you-
really don't.
They lost the genetic lottery.
Yes, they did. Yes, they did. I will say, in terms of the genetic lottery, no one in my family that I am aware of has peanut allergy. As a matter of fact, I can't think of any of us that have a serious allergy to food of any kind. We're all a little lactose intolerant, we're all a little gluten sensitive, but nobody's like, oh my God, if I eat that thing, I'm gonna die.
So is there a way based on like your family's genetics to determine if your kids would have allergies or have?
I am
more allergies?
sure that there is.
Well, yeah.
I am sure that there is.
Cause my sister has like every allergy under the sun.
I'm sure that there is, because allergy testing has become such a prevalent thing. Although I don't know if you guys have ever done allergy testing.
It's hell.
but it is not something that I ever would want to do. Cause, well, it's not even the needles. I don't care about the needles. I get tattoos. I'm okay with the needles. It's the fact that they're injecting the allergen. Do you not know how they do that? They make basically a grid on your back and each spot is a different allergen. And then they rate your allergic response based on how bad the reaction is in each of those spots.
I still have the picture from the last time they did, and it hurt.
Oh, I'm sure.
If you do like an allergy panel testing, like, what if you just randomly get anaphylactic? How do they determine?
I assume that they stay close with an EpiPen when they're doing allergy
Yeah,
testing.
they have them.
Yeah, I'm assuming that's the case. Yeah. Okay. Autoimmune diseases, they come in any number of forms, including type 1 diabetes, multiple sclerosis, lupus, my sister has, rheumatoid arthritis, which I have. My mother has sarcoidosis, which is an autoimmune disease that is similar to lupus and RA, but not the same. And they basically affect everybody slightly differently. You know, for me, my RA, if I don't take my medication regularly, my hands will get to the point that I can't like open and close them because it hurts just to move them. So we try really hard not to get there. A lot of it is genetic. We all started finding out about these autoimmune diseases when my sister got tested because she was having all kinds of weird issues. And she tested positive for the gene that indicates that you are susceptible to autoimmune diseases. And so we all went and got tested just to see where we were at. I was positive for the gene, my little sister was not. Lucky her. Not bitter. So, you know, it's genetic. There's a lot of heredity. Some of it is environmental, right? For me, if I supplement with things like turmeric and cinnamon, it helps reduce the inflammation, which reduces the overall effect that I have. So I have a lovely cinnamon, turmeric, blackstrap molasses oatmeal every morning. Really good, really good. Ooh, interesting. I'll have to try that. I tried it in my coffee because somebody recommended that, and I was like, no, that just ruined my coffee. That's not happening. So you know, there are a lot of environmental factors that can come into play. Stress is a big one, right? Stress will trigger a lupus flare up in my sister, as will apparently a Schwanga root, which says on the bottle, do not take if you have an autoimmune disease, which my sister did not read. She took it and had a massive lupus flare up. She couldn't walk, it was a mess. Now, because have you guys had evolution? Any kind of an evolution class? Yeah. Okay. So there is this thing that happens in the world where every time an individual develops a defense against a pathogen, that pathogen will then evolve some way to get around that defense. And then the individual has to develop a new defense. And then the pathogen will develop another way around that defense. Okay. The antigenic variation, the differences in the antigens for a particular pathogen, okay, will allow for this immune system avoidance. Basically what that means is, so antigenic variance is to a virus like blue eyes, brown eyes, and green eyes are to humans, right? It's all eye color, there's just variation within that trait. With the antigen, variation within that antigen can allow for avoiding the immune system. Think about antibacterial hand sanitizer. Okay, I don't use antibacterial hand sanitizer. I will use regular old soap and water perfectly happily. It does just fine, but I don't use the antibacterial hand sanitizers or the antibacterial soaps, because what do those bottles all say? 99.9% effective. That 0.1%, those are the ones that are able to avoid and survive that antibacterial stuff. That's the population that is left to make more. So we're just growing superbugs.
Yo,
Uh-huh.
yo, that like blew my mind. What?
Well, that's why, so there was a point in time, it's amazing to me that people don't understand. There was a point in time that penicillin killed everything. Now penicillin kills very little, right? All of those bacteria have evolved to be resistant to penicillin. Cipro is about the only antibiotic that we have now that is relatively able to kill most things. They very rarely give Cipro because if you give it to, so the reason we have all this antibiotic resistance is because for a long time, doctors would prescribe antibiotics just to shut patients up. Seriously, you would go to the doctor with the flu, and the reality is with the flu, there's not much they can do, right? You go home, you eat some soup, you drink some water, you sleep, and your body will kick it eventually, right? But patients would complain. Why aren't you doing anything to help me? Okay, fine. Here's a prescription for an antibiotic. Not going to do a damn thing for you. Right. And that's the thing, right? So they're giving out all of these antibiotics. The antibiotics are getting prevalent out in the environment. The pathogen is learning or is evolving to be resistant to that antibiotic. And so there's this issue that we've now created where there are methicillin resistant bacteria. You know, MRSA is a real thing. And it's a problem because we don't really have anything that can kill it.
Is that also why you have to keep all the antibiotics that you prescribed to?
Yes, because if you don't take all of the antibiotics in your bottle, you've basically killed off all the really weak ones and left the stronger ones behind. Yeah, gotta finish the antibiotics. And then you gotta take a probiotic because the antibiotics will kill everything.
Yeah.
Your gut bacteria just gets wiped all to hell and back.
Yeah.
So, all right, yes.
You think that was like unintentional or intentional?
Oh, it was absolutely unintentional. They didn't know what they were doing at the time. They know now. It's why it's so hard to get like a Z-Pack. They don't work. The Z-Pack doesn't work. My doctor has now started prescribing the bottles instead of the little 7-day because they don't work. You just come back for a second one.
Is it them unintentionally doing that is kind of similar to how like back in the day, they would just, if they found a disease that they thought was uncurable, they would just put them in the mental hospital and say, to hell with it.
Mm-hmm. Similar.
Lovely.
Yeah.
From your perspective, would you say it's not like a life or death?
You got that dog in you, bro.
So me personally, I am a throw everything but the kitchen sink at it kind of girl. If I get a cold, I'm taking all the cold medicine, like all of it, because I don't have time to be sick. Yeah. Yep. It's cough syrup right out of the bottle. Put me out, man. I don't have time to be sick. I need to get over this. Yeah. Now, is that the healthy way to do things? No. But it was a competitive powerlifter. We don't do things that way.
You don't have time to stop.
Right? I don't have time. I've got too many things. right? I had one semester, and this is what happens, right? You redline your body. I had one semester, it was the fall, I want to say it was the fall of 2019, and it might have been COVID, I don't know. But I was sick for like a month and a half. And there was like a week and a half period where I literally had no voice because I had coughed so much that I physically could not speak. I had to cancel classes.
Yeah, wasn't that around the time when they
Yeah.
were saying, like, it's the
Yeah.
vaccine food virus?
Mhm.
And then it just was like,
Yeah.
wait, it's mutating a little too much.
Yeah. Yeah, it might have been COVID, but yeah, I don't normally get that sick. Normally it's like a couple of days. I figured out a long time ago that it's better for me to take a day off and stay home and rest than to try to push, because then it just delays it. So
It's because at that point, your body is essentially requesting for you to sleep.
oh, my body doesn't request, my body demands. Sit your ass down.
If only I could tie you down and keep you there.
Right. Okay, so this is just a table that compares innate immunity to adaptive immunity, and then within adaptive, antibody versus cell mediated. It's just a summary table, right? All of the things we've already talked about, they're summarized here for you.